All right, so last time we talked a little bit about the history of how DNA was discovered. We touched on that a little bit. Today we're going to get into the heart of what DNA actually looks like. So DNA and RNA as well are both what are called nucleic acids. Remember we talked about it way back at the beginning of the year, we talked about proteins, carbohydrates, lipids, and nucleic acids. These were types of organic compounds. So DNA and RNA are nucleic acids. And the building blocks of both DNA and RNA are what are called nucleotides. So a nucleotide consists of three things. And this is actually true of DNA and RNA. They're both made of nucleotides, but they are still slightly different. So we're going to talk about DNA first. So a DNA nucleotide has what's called a nitrogenous base, a 5-carbon sugar, and a phosphate group. In fact, RNA has these same three things. What differs is the nitrogenous bases, so they are a nitrogenous base, they call it a base. I have a better picture coming up. There are 4 of these in DNA. They're called A, T, G, and C. Adenine, thymine, guanine, cytosine. You may have learned about these before. When you actually draw them out, they look kind of like these ring structures, and there's nitrogen in them. So that's one way you know that it's a nitrogenous base, is you'll actually see nitrogen atoms as part of the structure of them. This is a very simplified version. The 5-carbon sugar in DNA specifically is a specific sugar called deoxyribose. And that's where the name, the D, in DNA comes from. In RNA, it's not deoxyribose, it's a slightly different sugar called ribose. And then both DNA and RNA, they both have a phosphate group. And this is also simplified. Phosphate doesn't actually look like this. It actually is a phosphorus with 4 oxygens around it. Some of them have little charges on them. But they usually simplify it. Sometimes they'll put a letter P with a circle around it, and it's sort of a simplified way of representing phosphate. So this is one nucleotide, and a nucleic acid, like DNA or RNA, would have many, many, many of these nucleotides all joined together. And the way that they connect, they make a backbone of sugar and phosphate. So what you'll see in DNA is you'll see sugar and then a phosphate, and then the next sugar, and then the next phosphate, and then the next phosphate here, and then a sugar, and it would keep going. Specifically, these are the 4 nitrogen bases. Each nucleotide can have a different one, and we represent them with simple letters. So adenine we represent with A, cytosine is C, thymine is T, and guanine is G. Okay, so the full name of DNA is deoxyribonucleic acid. So the nucleic acid tells us it's in the nucleus. That's where we find it. The deoxyribo part is the name of the specific sugar. See how RNA is ribonucleic acid. So the sugar in RNA is going to be ribose, whereas DNA has deoxyribose. And there is another difference. Remember I mentioned that DNA had these 4 bases called, the nicknames for them were A, T, G, and C. Well, it turns out that RNA, and we'll talk more about this later, does not have the base T, thymine. It instead has a slightly different structured base called uracil, which we represent with a U. Now, RNA is also usually single-stranded. DNA, you probably know already, is usually double-stranded, meaning it's a double helix. It's got 2 sides that connect to each other. RNA usually doesn't. RNA is usually just a single side, like this, and there's not a second side connecting to it. RNA is actually, in a lot of cases, temporary. There's a kind of RNA called mRNA, and we make it when we're going to make proteins, and then it gets broken down when we don't need it anymore. There's another one called tRNA, which is actually just little nucleotides on their own. But DNA is very stable. It's this double helix structure. And this is actually a better drawing to show you in more detail. So this is deoxyribose. This is the phosphate group. Notice they actually drew out the whole phosphate molecule with all the little oxygens around it. And this is a nitrogen base. See how the nitrogen bases actually have nitrogen in them? And this happens to be thymine, a particular one. And the sugar deoxyribose, you're not going to be tested on this, but fun fact, a difference between deoxyribose and ribose is right here on this carbon. Notice how in deoxyribose, it's just a hydrogen here, but on ribose in RNA, it's an OH. there's an extra oxygen, and that's actually where the name deoxy comes from, is that deoxyribonucleic acid has the sugar deoxyribose, which is actually missing one of the oxygens that's present in ribose. This is in RNA, this is a nucleotide of RNA, and this is actually uracil. You're not going to be tested where I'll show you like a picture of these two, and you have to know which one's thymine and which one's uracil. I would not expect you to know it on that level, but you can see that uracil is just slightly different. Right here it has a hydrogen, and this has a methyl group. But that's not something we're going to get into the details of. Just know that in general, DNA has deoxyribose, RNA has ribose, and that one of those nitrogen bases is different. In DNA, it's called thymine. In RNA, it's a base called uracil. Now, it turns out those four bases are different from each other. If the base consists of a single ring, so see how thymine, it's just one ring, one hexagon, and cytosine also one hexagon? Those are called pyrimidines. So it turns out that thymine and cytosine are pyrimidines, C and T. A and G, adenine and guanine, are actually called purines, and purines have two rings. It's like a double ring, like two rings sort of fused together. And you'll see a picture of this later when we see how all of this comes together. Now, you're not going to be tested on knowing which bases are purines and which bases are pyrimidines, but little preview, in AP Bio, they do have to know the difference. And one of my students had come up with a cute way to remember this. They always remembered angels are pure. And that way they could remember A and G from angels were the purines. A and G are your purines. And they also remembered that angels have two wings, just like A and G have two rings. So two wings, two rings. So I thought that was kind of a cute way to remember purines versus pyrimidines. So here's what it looks like when you put it all together. This is one side. And this is that sugar phosphate backbone. So see how you have sugar, phosphate, sugar, phosphate, sugar, phosphate. And then here's the bases, the nitrogen bases. And again, see how they're actually these ring structures. This one has the double ring. This happens to be guanine. This has the single ring. And then when you actually put them together into a DNA, you see how the sides of the ladder are the sugar phosphate backbones. And the middle of the ladder, this is your nitrogen bases connecting to each other. And they actually connect, it's going to come up later, but they actually connect by very weak, what are called hydrogen bonds we talked about at the very beginning of the year. Okay, so a little bit more history. So remember Hershey and Chase, they were the ones that figured out that DNA was the genetic material. And then there was this big race. There's actually a movie about it called The Race for the Double Helix, where they were trying to figure out the structure of DNA. And the Nobel Prize was actually awarded to these two guys, Watson and Crick. There's a lot of controversy about it because all they did, honestly, was play with Tinker Toys. They basically took all the information. So poor Rosalind Franklin was doing X-ray diffraction. She got cancer from the X-rays, and Wilkins was working with her, and these other people were working with pneumonia. And then Watson and Crick basically went in a lab, and they took out a bunch of Tinker Toys, and they put them all together on a table, and they figured out, smart of them, but they figured out the 3D model. They figured out how the DNA strands went together into a double helix. And then they won the Nobel Prize using everybody else's hard work. So a little controversial. But ultimately, what they figured out was that hydrogen bonds held the bases together in the middle, and they figured out that A always connected to T, and G always connected to C. So it's always one purine and one pyrimidine, which makes sense because remember how purines were the double rings? So they're kind of bigger. And so it's always going to be one of those bigger ones connecting with one of those smaller ones. If they didn't connect that way, DNA wouldn't look like this. It wouldn't be straight. It would have like bulges. Like if you have two double ringed bases connecting, that's going to make it bulge out. This was also based on another guy's work, Shargaff. He actually did some experiments and figured out that the percentage of A and T were equal, and the percentage of G and C were equal when you analyzed the nucleus of all different kinds of organism cells. And so they also used his information. Again, he did all kinds of analysis and whatnot. He wasn't playing with Tinker Toys. But they took his information and the X-ray diffraction information and the pneumonia mice experiment information and the virus information, And they built this model using all of that information. So Chargaff, his rules said that the amount of A and T were always equal in cells, and the amount of G and C were always equal. And basically Watson and Crick took that information and figured out, oh, well, if A is always equal to T, maybe that's because A and T connect to each other. And that's exactly what they did when they built their model. They figured out that A connected to T and G connected to C. And so here's your sugar phosphate backbones that we talked about. Here's our bases down the middle. And A, if you know one side, you can figure out the other side. If you knew this was a C, you know this has to be a G, because C can only connect to G. Notice, again, that it's always one of those bigger ones. They're representing them here. This is a purine. This little one's a pyrimidine. It's always one purine and one pyrimidine, because if two of these got together, the DNA would be wider at some points and skinnier at other points. And again, this middle, they're connected by hydrogen bonds, which are usually represented by dotted lines. And that's important because hydrogen bonds are easy to break. So when DNA needs to make copies of itself and things like that, it's really easy for it to unzip in the middle. The bonds on the sides, by the way, are covalent bonds. We've talked about those. Those are very strong bonds. So these are not normally breaking. The only thing that can break easily are these hydrogen bonds down the middle so that DNA can open up. And it needs to open up for replication as well as for when it's doing its job, which is coding for making proteins. This is just showing that twisty double helix, twisted ladder shape. And again, this is showing a practice. So if we know one side, you should be able to figure out the other side. So we know A only connects to T, so this has to be a T. T connects to A connects to T, T connects to A. See how easy this is. Should do a whole test of just these. C to G, A to T, T to A, C, G, C, C, C. Notice how they separated these in threes? That's going to be a little preview for something we're going to learn later. All right, so that's kind of an overview of DNA structure. Ooh, sorry. This is a last little part of the history. So Rosalind Franklin, this is her experiment. So she did something called X-ray diffraction, where she bounced X-rays off of DNA. She died before these two guys won the Nobel Prize. I believe of cancer, I believe because of radiation exposure from her experiments. But she actually figured out it was a double helix. Whenever you bounce X-rays off of a molecule and you get this X-shaped pattern, it means helix. So basically, she's the one that figured out that it was a helix. Chargaff figured out that A was equal to T and G was equal to C. Wilkins worked with her, Maurice Wilkins. And bottom line, Watson and Crick, these two guys, they actually won the Nobel Prize, and this is the model that they built to do that. All right. And that's it. That's just another overview. See the hydrogen bonds, the structure. If we give you one side, you should be able to figure out the other. Make sure you know that the sugars and the phosphate form the sides. Also, these two sides are something called antiparallel. It may come up a little bit later, but see how one side is upside down compared to the other, how the oxygens face down here and they face up here? That actually is important for when DNA copies itself. The idea that one side is sort of running in the opposite direction of the other. But it'll come up when we talk about this next section on DNA replication.